Alright, let's take a step back and see how this messenger RNA came from DNA. Keep in mind that the translation start site, which is the AUG codon right here, is not the same place as the transcription start site, which is the beginning of the 5' untranslated region. And the same is true at the other end. There's a lot of stuff that's translated into mRNA that will not be in the protein, which ends here. What are these untranslated regions for? They actually have a lot of important functions, like they have instructions for modifying the ends of the mRNA to add the 5' cap and the poly A tail, and the 5' UTR has basically a landing strip that guides the ribosome to dock on the mRNA. There are also other sequences within these UTRs that are important in regulating where the mRNA ends up in the cell and how long it lasts. Okay, so that's the mRNA. Now the genes, as in DNA, have a few parts to them as well. At the 5' end, there are promoters, enhancers, and silencers. These are just sequences within the DNA, not other types of molecules. Proteins like transcription factors can recognize these sequences and bind to them to activate transcription in the case of promoters or enhancers, or repress transcription when they bind to silencers. Generally, promoters are within a few hundred base pairs of the transcription start site, whereas enhancers and silencers can be tens of thousands of base pairs away and can also be found in the gene itself, mostly in the introns. One of the most important sites is the TATA box, which is an area about 25 nucleotides upstream from the transcription start site in eukaryotes. Why is it called the TATA box? Because it's a sequence of mostly T's and A's. Now, if you remember the beginning of the lecture, is a sequence of lots of T's and A's going to melt apart easily, or are these T's and A's going to hold tightly to each other? So the answer there is they're going to melt apart easily. And this is important because the TATA box is where the proteins involved in transcription begin assembling, since they can easily separate the two strands of DNA here. In fact, the first protein to start the process of transcription is called the TATA box binding protein, or TBP. It can bind here and recruit other transcription factors. There's also a similar sequence called the CAT box, which is about 75 base pairs upstream from the transcription start site, and this also helps regulate transcription. Okay, so the transcribed region, which is this here, is made up of introns and exons. Which have the part of the mRNA that will get translated? Right, that would be the exons. The exons are the part that will actually make it into the final mRNA, and the introns will be removed by a process called splicing. Once again, the transcription start site here, which will be the beginning of the mRNA, is different from where the start codon is, since the start codon is the start of translation, and it codes for the first amino acid of the protein. If we compare this DNA image to the RNA image above, transcription begins here, and then we have our 5' untranslated region, and the start codon is further down. Another thing I want to point out about this diagram is that the 5' cap and 3' poly A tail are added later, and were not part of the original DNA sequence. Okay, the last thing for this slide, make sure you keep in mind the difference between the template strand and the coding strand. Remember, we have our two strands of DNA that are complementary to each other. Which strand is more similar to the mRNA, the template strand or the coding strand? Well, the template strand is read by RNA polymerase to make the RNA, like as its template, so it's complementary to the mRNA. The coding strand will have almost the same sequence as the mRNA with two exceptions. Do you know what those are? The mRNA is only different in that it has uracil instead of thymine, and it has the intron spliced out. And to review from earlier, if mutations occur in promoters, it can change the amount of transcription of that gene, usually by reducing it.